Hi, my name is Benjamin Guthrie. I'm a recent graduate from UC Berkeley out of Michael Marletta's lab. I'd like to thank the organizers for inviting me to present my work in this format and you, the audience member, for tuning in today. I'm excited to tell you one story from my PhD, and the title of my talk is 2 against 10,000, the structure of the mammalian nitric oxide receptor. The Marletta lab is interested in the different roles that nitric oxide plays in human physiology. One of those roles is in your immune system. If you were to walk into a chemistry lab and see a tank of nitric oxide, it would definitely have a poison gas label on it. Indeed, our innate immune system uses nitric oxide for this very purpose. Macrophages can secrete a high concentration of nitric oxide for long amounts of time to kill invading pathogens. The enzyme that's responsible for this phenotype is a nitric oxide synthase, in this case inducible nitric oxide synthase, which catalyzes the 5-electron oxidation of arginine to citrulline and nitric oxide. However, there is also a non-toxic role for nitric oxide in human physiology, and this is in your vasculature. Nitric oxide acts as a signaling molecule, the end result of which is to dilate your blood vessels and lower your blood pressure. The enzyme that's responsible for this is still an nitric oxide synthase. This is endothelial nitric oxide synthase, which has a lower output of nitric oxide. However, this lower output of nitric oxide is sensed by a very specific receptor, soluble guanylate cyclase, or SGC. Let's take a closer look at this signaling pathway. The nitric oxide SGC signaling system helps to regulate vascular tone. The system is initiated in endothelial cells, where influxes of calcium activate nitric oxide synthase to produce nitric oxide. Nitric oxide is a diffusible gas that can diffuse across plasma membranes into the smooth muscle target cells. Once here, nitric oxide binds the SGC, activating the enzyme. This increases the concentration of cyclic GMP in the cell, which in turn regulates cyclic GMP-dependent kinases, phosphodiesterase, and cyclic GMP-gated ion channels. Because SGC is the first enzyme within the target cell, we really need to understand how the biochemistry or how the activation of this protein occurs. We know that with no nitric oxide present, SGC is not active. Even with just a stoichiometric amount of nitric oxide, the enzyme is still only marginally active. It takes either excess nitric oxide or one nitric oxide with a small molecule stimulator to reach maximal activity of soluble guanylate cyclase. Bayer has put a lot of work into developing these small molecule stimulators, and in 2013, Adempas became the first FDA-approved SGC stimulator. This is used to treat certain forms of hypertension, pulmonary arterial hypertension, and chronic thromboembolic pulmonary hypertension. But how these small molecules and diatomic gases activate SGC is still not understood. I want to introduce the different domains of SGC. SGC is a heterodimer composed of an alpha and beta subunit. Each subunit is divided into 4 different subdomains. At the N-terminus, the first is the HNOx domain, or the heme, nitric oxide, and oxygen binding domain. In SGC, only the beta HNOx is competent to bind the heme cofactor, and this is where the first nitric oxide molecule binds. Next, we have 2 sets of dimerization domains. First is the per-arndt-sim or PAS-like domains. Next comes the coiled coil domains. Finally, at the C-terminus, we have a catalytic domains, which is responsible for cyclizing GGP into cyclic GMP. We do have the structures of each subdomain of SGC. However, this cartoon that I have of the full-length structure down the bottom isn't entirely what it seems. Our lab has used negative stain single particle EM to get this dumbbell-shaped volume to which we map the individual crystal structures on top of. However, this doesn't have the structural information for the loops or the interdomain interactions that we really need to understand how the separate gas sensing and catalytic domains interact to activate soluble guanylate cyclase. Biochemical and biophysical experiments have led to different hypotheses for how the signal transduction occurs in SGC. The first looked at catalytic domains by themselves and adding different amounts of HNOx or HNOx and PAS constructs in trans. You can see that with increasing equivalence, the activity of the catalytic domains goes down. This led to the hypothesis that there is a direct interaction between the gas sensing portion and the catalytic portion of SGC. However, in the single particle negative stain EM analysis, no matter what the ligandin state was, there was always large amounts of motion between the particles. Thus, there are two different hypotheses for how the signal transduction mechanism in SGC functions. The first is the direct contact hypothesis, where there is a self-inhibition by the beta HNOx domain onto the catalytic domains. And the second is the through the protein backbone hypothesis, where the particle is continually moving around, maybe to catch all of the nitric oxide that is present. And then once nitric oxide is bound, then there is a conformational change along the protein backbone that activates the enzyme. So the questions that I want to focus on for the rest of my talk are: how are the domains of SGC organized in space? How is the nitric oxide signal propagated from the HNOx domain to the catalytic domain? And what conformational changes are necessary for enzyme activity? When I began this project, there was a new homologue of SGC that was shown to interact with drug-like molecules. This is a homologue from a moth, Manduca sexta. And so I began the structural project asking if it would be possible to obtain a full-length structure of either the moth or the mammalian SGC. First, I needed to express and purify moth SGC from insect cells, which had not been done before. Insect cells are our expression system of choice, not because of the highest yields, but because of the highest heme incorporation. Using a two-column system over a nickel iMac and a porous ion exchange, I was able to purify moth SGC to homogeneity and confirm with mass spec that I had the expected subunits. Next, I needed to make sure that Manduca sexta SGC behaved similarly biochemically to mammalian SGC. I did this through two different biochemical assays. The first is UV visible absorption spectra, where we track the ligand-dependent changes of the heme binding domain. Here at 433 nanometers is the Sorey band of the unliganded SGC. This blue shifts to 424 and 399 nanometers when carbon monoxide and nitric oxide are bound respectively. Next, I confirmed that the ligand activation profile of Manduca sexta SGC matched what was expected. So in white, we have Manduca sexta SGC without stimulator, and we can see that there is low activity with no ligand bound, some activity with one nitric oxide, but full activity with excess nitric oxide, and critically that the YC1 stimulator molecule did show this activation in the 1NO state. And this is similar to what we'd expect for the mammalian version: little activity with nitric oxide, and then full activity with excess nitric oxide and 1NO plus YC1. While the individual subdomains of SGC have been amenable to structural determination via X-ray crystallography, the full-length structure has remained elusive. Thus, we turned to the recent advances in cryo-electron microscopy in hopes that we would finally be able to capture the full-length structure. Cryo-electron microscopy works by freezing protein in vitreous ice. This allows for clear structural determination by image capture and finally in silico reconstitution of the three-dimensional structure. Now, because of the cryo-EM detectors and the software that is used to solve the structural reconstruction, the larger and more ordered your protein sample is, the better. Unfortunately, SGC is 140 kilodaltons, and because of its elongated shape, it's really like a 70 plus a 50 kilodalton protein. However, I'm going to admit many months of troubleshooting. We did need to overcome a preferred orientation issue upon freezing, but we were able to solve the first full-length structure of SGC. All of this work is done with my great collaborator Adam from James Hurley's lab, also at UC Berkeley. And you can see here that I'm showing you a map of 5.1 angstrom resolution, so not quite good enough to get side chains, but definitely good enough to kind of get the overall structure. Here we have fit all 8 subdomains into this compact conformation of the inactive structure. Let's take a look at a movie of this map. So here we have the map of SGC. We can put the domains into this map. And let's go ahead and zoom in on the beta HNOx domain. So we can see here that we have a lobe of density that is not filled by the protein backbone. And this is where we believe the heme cofactor resides. So we do think we have hollow SGC here. Surprisingly, when we looked at the back of SGC, the coiled coil domains were bent, and we really had no prior knowledge of this. This is the first time that we had a view of this, and so potentially this could be important for the sealing mechanism. Next, down in the catalytic domains, we can look and see that this is in the closed catalytic dimer conformation, and we can see that because if we model in a nucleotide from an active adenylate cyclase crystal structure, there is a very large clash between the nucleotide and the catalytic domain. Excitingly, we were also able to resolve a map of active SGC. So this is at 5.8 angstroms. We were able to fit 7 of the 8 subdomains into this map. We were not able to resolve the alpha HNOx domain. Let's take a look at a movie of our active map of SGC. Here is our 5.8 angstrom map. Again, we were able to fit 7 of the 8 subdomains into this map. We were missing the alpha HNOx domain, which is probably not very ordered. In the beta HNOx domain, we have an extra lobe of density. This is where the heme cofactor binds. And now as we swing around to the back of the enzyme, you'll see that the coiled coil domains are now straight. So they were bent in the inactive form, and now they're straight in the active form. We also saw this new surface between the beta HNOx and the beta coiled coil domain. And when we swing around to the catalytic domain, we can see that this cat domain is now in an open conformation. We can now easily model in a nucleotide into our map, suggesting that the enzyme is indeed in this active conformation. Now that we have the conformations of both the inactive and the active form of the enzyme, we can look at a morph between the two. You can see that as this morph plays, we have this huge 74-degree rotation of the gas sensing part, the HNOxes and PAS bundle, and this is going to pull on the coiled coil domains, which in turn will reorient the catalytic domains relative towards each other. We're holding this alpha catalytic domain constant here. So this either push or pull or whatever the conformation, the impetus for the conformational change is, this is really going to realign the catalytic domains. Here is our new interface that I mentioned, and we also in the active state noticed an extra lobe of density here. And so if we zoom in here, we can see that the heme is placed far away from it, and we think that this is where the small molecule stimulator binds. I'm going to swing around the protein for you to see, so you can see that the YC1 stimulator binds right in between the beta HNOx and the 2 coiled coil domains. We think this is really exciting and think that this molecule acts kind of like a wedge to help the beta HNOx and this new interface form in the active state of the enzyme. We were really excited to see such a large conformational change between our inactive and active conformations of SGC in our EM data. But it made us wonder if we could see the same conformational changes in solution as well. For this, we turned to small angle X-ray scattering, or SAX. In SAX, you shoot an X-ray beam at your sample, and then you measure the angle at which those X-ray beams scatter. If we were to look head-on at this X-ray detector, it would look something like this. And then if you radially integrate from large angles to small angles, you get this scattering curve like this. The scattering curve has lots of information about different properties of the molecule, and SGC scattered very well. We have a nice radius of gyration, volume, and the molecular weight was agreed with what we expected it to be. We were able to take advantage of an inline UV vis detector on our SAC sample to determine the ligand state of our SGC. On the left, I'm showing you a UV vis spectra of Manduca-Sexa SGC, and what we're going to be looking at is just these two wavelengths. So here is our unliganded sample. You can see that the ratio of the unliganded at 280 to unliganded at 432 is approximately the same as the ratio of these two peaks here. When we added NO to our sample, these were the peaks that we saw. And you will notice that 1NO has a slightly higher absorbance than the XSNO sample. And from this analysis and other normalizations that we did, we were able to show that because of the technical limitations, things had to be anaerobic and such, we were only able to get the 1NO state of SGC in our SACS data. The scattering of Manduca SGC showed that we had a well-behaved particle, and it was folded as well. And we were able to look at the extension, so the longest dimension of the particle by SACS. And so from this, we were able to show that the 1NO sample of SGC was partially extended. We had a 100% compact conformation when there was no gas present, but only 72% compact and 28% extended for our 1NO conformation. Here are the fits to those models. Thus, we have a structural model for SGC activation, and this has three distinct states. In the inactive state of SGC, we have a compact particle. When you add the first NO, we see that minimally active state, and we think this is because there is a conformational equilibrium between 72% inactive and 28% partially extended, and it's the partially extended portion that is giving the small amount of activity. Then, if you add XSNO or YC1, you get a maximally active, fully extended particle of SGC. That's responsible for the activation of this signaling pathway. While our manuscript was in review, another cryo-EM structure of SGC was published, and we were pretty excited to see that our structures overlaid pretty well, especially because the other group did not use the small molecule stimulator, they only used XSNO. I will say that their structure is of higher resolution than our structure, and we can use their high resolution structure and our location of the small molecule stimulator to further improve these class of drugs. Neither structure of SGC found direct contact between the beta-H-Nox domain and the alpha-catalytic domain. Thus, both structures are in support of the through the protein backbone hypothesis, where there are interdomain interactions between amino acids that transduce the signal from the gas sensing portion of the protein all the way down to the catalytic domain. To conclude, I've shown that SGC possesses a bent coiled coil region when it is inactive. These coiled coils act as a hinge to convert the protein from an inactive form to an active form. And finally, that YC1 small molecule stimulators of SGC bind in a central location between the heme cofactor and the coiled coil domains of SGC. In the future, there needs to be more site-directed immunogenesis now that we have a higher resolution structure of critical regions to probe activation mechanism. It would be very interesting to obtain cryo-EM structures of other activation states, including the 1NO states. And Whether the resting state of the enzyme is actually in the compact conformation or the extended conformation, we don't know yet. So it'll be interesting to conduct biophysical experiments to see what forces are involved in SGC. Finally, I would like to thank my mentor, Professor Michael Marletta, for all his support and guidance over the course of my PhD. I've learned so much about science and being a scientist because of you, and I can't wait to keep collaborating in the future. Also, to my lab mates, thanks for making the Marletta Lab such a great place to work. All the wonderful conversations and times we've had, I'm going to really miss that. Also, to my collaborators, Professor James Hurley, Kyle Morris, and Adam Yoakum, I couldn't have done this without you. Thank you so much. Also, thank you to Michael Hamill and Daniel Rosenberg for help with the SACs. These sources for funding, and all of you for your attention. Thank you very much. You can find me on Twitter at BGHorst, and I look forward to engaging with you there. Have a great day.